Okay, so thank you so much to Katie, to Liz, to the Lily Foundation. I feel enormously privileged. It's an honour to come and speak to you this evening about this topic, and I'm also grateful to so many people giving up their Tuesday evening. I'm sure they must have better things to do, but anyway, nice to see you all. The title I've been given by Katie and Liz is Mitochondrial Disease Research, Progress, Promise, and the Honest Reality of Treatment Development. So I think the one thing I'm going to definitely try and do is be honest with you all. I'm sitting in my office in the Institute of Child Health at UCL. I'm just around the corner from Great Ormond Street Hospital, well, next door to it, and around the corner also from Queen Square and the Institute of Neurology. And it's a really historic place. It's where the first mitochondrial genetic variants causing disease were discovered, gosh, nearly 40 years ago, 38 years ago. And I came here a few years after that and started seeing children. Some of the first patients diagnosed with mitochondrial disease genetically, but it was a really challenging time. We were able to find the genetic cause in fewer than 5% of people who were thought to have mitochondrial disease, and there were no treatments and little in the way of support. And I am really pleased that particularly in the last decade, we've seen remarkable progress in the mitochondrial disease research field and clinical care. And one of the biggest breakthroughs has been in genetic diagnosis, and that's because we've got new sequencing technologies called next generation sequencing. And these have meant that we can get an answer for about 50% of people on routine testing, and perhaps you can get that up to 70% with research testing. And I just want to show you a little snapshot of what this looks like in reality. So this is using something called whole genome sequencing in a child who's very unwell, and you look at the child and their parents together, and the parents are being controls in that setting. And you can get a diagnosis in just a couple of weeks, perhaps even less. And I'm going to show you a real example. It's not this year, but it was a year when a sample was sent off on the 23rd of May, and we got an answer there on the 8th of June. So that's really very quick, isn't it? And that's compared to when I first came here. If we were able to get an answer at all, and particularly most of the time it was through research, we could have been going back to people decades after their illness onset. So there is rapid progress. And I think that if you'd asked me back then if it would ever be possible to sequence the genome, I would have said, no, I can't imagine it, and not at anything that was a reasonable cost, which it is now. And therefore, I want to say to you that even if things don't seem fantastic on the treatment side yet, things are moving so fast that I think we are likely to have treatments, a lot of treatments in the next decade, I hope. And I hope I'm not proved wrong there. And just the last thing to say about this type of genetic testing is that it can be initiated in a general hospital. There is no postcode lottery. It is NHS routine testing. It is not research. And it means that care can be directed to the right specialist as quickly as possible. But we're not there yet. That's why we're here this evening in conversation. We still lack curative treatments for the vast majority of people affected by mitochondrial disease. And that for me is a devastating thing to have to say to you, particularly when I've been in the field so long. And I really, really hope that this is going to change. And the question then, of course, is why is it so difficult to treat mitochondrial disease? And for many of the things I'm going to say today, there isn't a single answer. So we'll start with the first answer to this. The first thing is, of course, it's not one disease. It's a large group of disorders with different genes affecting different organs in different people, different ages of onset, and different rates of progression. Mitochondrial diseases are complex. This is a cartoon that I made with a journal a few years ago, just showing that really any organ or combination of organs can be affected with any onset of age, and this can happen at different rates in different people. And what complexity looks like in real life is this. These are images of people from published literature who have mitochondrial disease. And you can see that some people might have a characteristic appearance and others don't. It can affect hearing, it can affect the heart, the hair. There can be extra hair. The EEG can be abnormal. The blood film can be abnormal. The optic nerve can be very pale there. There can be bony abnormalities. There can be extra pigmentation in the retina, the back of the eye. And back in the day, we used to do muscle biopsies to diagnose mitochondrial disease, and these ragged red fibers and Cox negative fibers were our diagnostic clues. And on the brain scan, we can see stroke-like episodes, sadly, in some people. So complexity is challenging, and we now know that more than 450 different genes can cause mitochondrial disease. And I think it would be naive to think there's going to be a single treatment that's going to cover all of those different genetic causes of mitochondrial disease. This is a picture from the Lilly Foundation, and all of these people have mitochondrial disease, and you can see that they have different problems. And different organs can be affected in different people at different times, and that also creates challenges in developing treatments. So I've said to you that there are many different causes of mitochondrial disease, and we may need different treatment approaches for each, and an individual may have a large number of different problems, and a single treatment may not be able to cure them all. Other challenges, well, the mitochondrion, as you can see here, let me just turn my pointer into a laser. There we go. Yeah, so it's got these two walls. There's this outside wall, and then there's this convoluted inner wall. So whatever we give in the way of drugs or genetic therapies have got to get through these two walls, and this particular one is very difficult to penetrate. So that's another challenge. Another one is that the way that drug treatments are developed is that often you have to do animal testing. And until recently, we didn't have very many animal models of mitochondrial disease in which we could test drugs in what we call preclinical trials. And I'll come back to mice a bit later on. And then a thing that I want to talk about a lot is that even if we overcome all of those things, a big problem is difficulties in clinical trial design for these rare diseases because of the complexity and the heterogeneity that I have told you about. Okay, so we've talked a little bit about the challenges. It's not all bad news. The really good news is that there are lots and lots of potential treatments of mitochondrial disease being actively investigated, so many that if this talk was about those, we wouldn't have enough time to discuss them all today. But I'm just giving you some examples here. There are vitamins and cofactors. There are things that increase making new mitochondrial, what we call mitochondrial biogenesis. There are things that affect the redox state of the mitochondria. There are things that stabilize the mitochondrial membranes. There are things that affect something called mitophagy. And there are nucleosides, which I will mention again later, and also different kinds of genetic therapies. So that is good news. But how can you prove that one of those treatments, those candidate treatments, works in real patients? And that's what I want to discuss for the next few minutes of this talk. So that is where clinical trials come in. And if you've never been in a clinical trial, this might all be new to you. And for those who have been in it, apologies, go and make a cup of tea and come back in a few minutes. So what are clinical trials? They are research studies that test new treatments in people to see if they are safe and effective. You can test new medicines, other therapies, or medical devices even, and they follow very strict safety protocols and regulations, and the participants are carefully monitored throughout. It is quite a burden to be in a clinical trial, but these are incredibly important, and they help to determine if the treatments can be approved. So this is a cartoon that I've got from the web. You've got the preclinical research, finding a possible treatment. You might have been looking in cells from patients or in animal models or something here in culture. And then you go through phase 1 to phase 2 to phase 3 studies. You go through approval by the regulatory agencies, and then eventually you get through to phase 4, which is where you're collecting data in real world use of those drugs. So let's break this down. Phase 1 is asking the question, is this treatment safe? Phase 2 does it work? Phase 3, is it better than our standard of care, the current options that we have? And then phase 4 is the long-term monitoring after approval. And that's also important because sometimes the clinical trials themselves, although they may have been done on a lot of patients in lots of centres, they may not know that there is some incredibly rare side effect that happens, that comes to light after many more patients have been treated over a longer period of time. I think an example I can think of is that, I can't remember which one, but one of the COVID vaccines, there was a very rare side effect that only came to light after the trials had been finished and the vaccines were being rolled out. Okay. So we're talking about rare diseases here, and rare disease trials are different. They have to be different. It's because fewer patients are available. Even if you go worldwide, the patients may be geographically spread out, so you can't do a trial in one centre. And for rare diseases, and especially where they're heterogeneous and complex, as mitochondrial disease is, you need every patient's participation to get the information that we need. And the flip side is that the regulatory agencies, and they're doing this increasingly, they're allowing more flexible approaches, not so rigid about how we do this. And I'm hoping that this will only get better over time. So I'm just going to show you in a few slides the difference between a common disease trial and a rare disease one. So with common disease, I'm thinking about things like treatments for heart attacks or Alzheimer's or diabetes. Even some cancers are common, a lot of them are rare. So for common disease, phase 1, safety testing, you go to healthy volunteers, maybe as many as 100 healthy volunteers before you'd ever go near a patient. And this is where I think students often get paid to go and do these trials, don't they? And the idea is to see if the treatment's safe, are there side effects, and what are the safe dose levels, and they last a few months. For rare diseases, you're not going to get that many patients. You actually go into patients, not into healthy volunteers often. And so it's between 5 and maybe 20 patients with the actual disease, testing safety in real patients, and looking for early signs of benefit, which you might call efficacy. And often, for rare diseases, the phase 1 is combined with phase 2. So let's go on to that. So here we have phase 2. For common diseases, you're looking at patients, maybe up to 300 with a disease, and it's the first phase of looking to see if the treatment actually works, looking for effectiveness and side effects, and this may go on for a couple of years. For rare diseases, you've got far fewer patients available, and I think it might be more likely 20, maybe as many as 100, depends on which rare disease. And you're gathering, again, evidence of effectiveness, clinical improvement, safety, and importantly, alternative trial designs might be used for rare diseases. So the classical trial design is placebo-controlled, double-blinded, randomized trials. So there you'd have one group that's getting the treatment, one that's getting placebo, and everyone's blinded. The patients are blinded, and the doctors administering it are blinded, because we all can want things to work so much that we can see improvements that aren't really there. And so alternative trials, I'll talk a bit about those a bit later on. Let's move on to phase 3. So these are the confirmation studies. You've got a positive signal from your phase 2, and it's worth the drug company, and often only drug companies can afford to run these trials. They're very, very expensive, and I think that academia, clinician-led trials are very difficult to do because of the expense associated with those. So for the common diseases, you may get thousands of patients. So the classical heart attack trial, I think it's called the Framingham study in America, that had, I think, 4,000 patients in there. And that was even 40 or more years ago. And what you're aiming to do is showing that the treatment that has got a signal in that phase 2 trial is working better than the standard of care. And these are the randomized controlled trials, and they may last several years, and they may be multi-center. For rare diseases, I think you're talking more like 50 patients. You might get to 300, depending on the disease, and availability limits that. And you're looking to show clinical benefit, and you may compare to placebo, or increasingly, for rare diseases, we're looking to see if we can use the natural history. That means what happens in the disease without a treatment. And that's why we're doing a lot of natural histories for mitochondrial disease, and I'll mention those later on. And often we're having to go global, international multi-center studies for these. So once you've done your phase 3 study, what you're hoping is that your phase 3 study has provided the evidence for regulatory approval, and then the places that could approve it in the USA, there's the Food and Drug Administration, in Europe there's the European Medicines Agency, and in the UK we've got the Medicines and Health Care Products Regulatory Agency, and NICE, the National Institute for Health and Care Excellence. And they have to weigh up all of the evidence generated from all of the trials. And I think the documents that pharma companies may be submitting to these agencies may run to thousands and thousands of pages from the trials. And they weigh up the evidence and they make a decision. And if that is successful and the drug is approved, then for both common and rare diseases, it doesn't stop there. Still need to carry on monitoring long-term safety and effectiveness and look for those rare side effects in real-world use, and this may go on for decades. So just to summarize what I've said to you, the key differences at a glance between common and rare disease trials is that small patient numbers in rare diseases make it difficult to recruit patients to the trials, and so it can be slow to recruit and it's much more difficult to prove benefit. What we are working towards is doing trials with smaller participant numbers. We talk about N of 1 trials, and flexible trial designs. There are things like basket trials, adaptive trial designs, and international collaboration is vital. We do need to work globally and not be insular and just looking inwards at our own centre or our own country, and that we may need to combine those different trial phases. Okay. That's what I want to say about clinical trials. I'm just going to have a sip of water. I'm moving on to say a few words about gene and cell therapy approaches of mitochondrial disease, because I know that Katie and Liz received a few questions ahead of this Zoom room about those. So this is where things get complicated, because we have two different genomes that can cause mitochondrial disease. There's the mitochondrial genome and the nuclear genome. And there are different approaches needed for each of those. But the mitochondrial genes approaches that have been taken include going in with things called nucleases, where you go in and selectively destroy the mutated mitochondrial DNA and leave the good mitochondrial DNA to take over. There's base editing, where you go and correct the mitochondrial DNA change. And there's allotopic expression, where instead the mitochondrial genome has its own code that's different to the code of nuclear genes. And this has been done for laboratory optic neuropathy, you take the mitochondrial gene that's got the mutation in the disease and you recode it with a nuclear genetic code, and you send it in to be expressed from the nucleus, and then the product sent into the mitochondrion. Except for the labors, all of this is very much at the preclinical stage. It's still in cell and animal models. Then there is mitochondrial donation therapy, which targets the mitochondrial genome, but this is not a genetic therapy, it's more a preventative strategy. And that has been successful in the Newcastle clinic, which is the only clinic licensed to do this globally. 8 babies were born, and the results were published in the New England Journal of Medicine last summer. The nuclear genome is slightly more straightforward. There's lots of other nuclear gene-encoded rare diseases for which genetic therapies have been done. And there are, again, a few strategies. The most common is where you send in a normal copy of the gene with a mutation, and by supplementing that, you provide enough of the product of that gene to cure the disease. And there have been successes in things like spinal muscular atrophy. And these use usually viral vectors to get the supplemented gene into the cell. There's also strategies using something called oligonucleotides, where you can go and where it's diseases, particularly say where there's one copy of the gene that's causing a problem and the other is normal. is healthy, it's what we call a dominant disease, and there are particular types of genetic variant that you can target by sending in these oligonucleotides that go and bind to the mutation and silence that copy of the gene, and the good copy is enough to take over the function. And then there are cell therapies, which include bone marrow transplants or liver transplants. I'm not going to say any more about these, but just to say there's a lot of stuff going out there. And if I just look at the nuclear encoded mitochondrial diseases, in mouse models, more than a dozen of them have been treated with these viral vector gene therapies, and all of them showed biochemical and clinical improvements. But the challenges are that there are very few mouse models of mitochondrial disease that are viable, so that survive into being born as mice and that replicate the human disease. And there's also, like I've shown you for the clinical trials, it's a very long pathway to develop the treatment. So we have to know what the natural history of the human disease is. We have to make and characterize a mouse model. We have to design the viral vectors and produce the virus in large copies to be able to treat the cells. And then we look in the mouse at a neonatal stage and an adult stage. And then you go to the clinical translation, which is very similar to the clinical trials I've talked about for the pharmacological therapies. And all of that can take 15 years. So that's a very long time. Why do these breakthroughs take time? Well, each step of all of these pathways that I've shown you exists to protect patients and to avoid giving false hope. And so one of the steps that I haven't mentioned is that we do only take things to clinical trials where there is equipoise. If you had a treatment that obviously works, then you wouldn't do a standard clinical trial and you might go straight to treating a patient. And examples I can give you for that are, say, some of the vitamin deficiency disorders. We know that you can't transport the vitamin into the body, and so giving the vitamin in high dose was obviously the answer. But otherwise, if you're not, don't have huge evidence that it's going to work, then we have what we call clinical equipoise, where if you had a set of scales, they're balanced. Maybe it's going to work, maybe it's not going to work, and so you have to do the clinical trial. And so we have the lab discovery, we have the safety work, we have the early trials, the large trials, and the approval access that I've told you about. But I have to say here, and remember we're talking about being honest here, something can look really exciting in a lab or even in a mouse and still fail in people. And this is common across all fields of medicine. And it's why careful clinical trials matter. So some trials fail, some succeed, all move the field. Negative trials are painful to experience, but if it is showing us that the treatment doesn't work, we would rather know that than not. So an example of a disappointing trial was the vatiquinone in the MIGHTY trial for mitochondrial epilepsy, and it didn't meet its primary endpoint. So that was very disappointing. And I think it's fair to say we don't really know if that might work in some patients for some indications, and probably more work will be done, and the company's probably planning that. An example of where trials succeed, well, elimipritide is one of those drugs I mentioned that stabilizes the mitochondrial membrane, and it was used in mitochondrial myopathies in adults from a large different set of causes, and the phase 2 trial looked positive. The phase 3 trial did not, and then they went back and they reanalyzed the data, and also for Barth syndrome, it's a type of mitochondrial disease that's affecting the mitochondrial lipids in the membrane. It made scientific sense that it might work, and so they did another trial of Barth syndrome, double-blinded, placebo-controlled, randomized, and did show efficacy, and that has been approved in the USA in 2025 to treat Barth syndrome. So the message here is that real progress in rare disease research means learning both from success and from failure. So some other real but limited treatment wins is for Leber hereditary optic neuropathy, LON. Idabenone, which is a modified version of coenzyme Q10, is authorized in Europe and the UK, and we're still navigating the NHS access pipeline. And then for TK2 deficiency, which is a type of mitochondrial DNA depletion syndrome causing a muscle weakness, I can't pronounce this, Kijevi, has just been approved by the FDA in the US, and the EU process is still ongoing. And that's to treat with nucleosides. So the Kijevi is the new name for the nucleosides. So these examples show that treatments are possible, but because these are only 2 diseases, they don't apply to most mitochondrial diseases. But it is showing us hope and that it is possible to get to approval. So this is a snapshot of the current mitochondrial drug development landscape and some of the drugs that are in development. So there's a KL133 in a phase 2. Gosh, drug companies and the names they give, how are we going to pronounce this? Zagosiguat? Zagosiguat is being developed for Miller syndrome by Descent, and that's in a phase 2b study. Sonlicromanol, I think I pronounced that right, by Chondrion for MELAS and MID caused by the 3243 variant in the mitochondrial DNA. And then a drug called PX578 by Pretzel. They are in phase 1, and unlike other drugs, because this had never been, it was a brand new compound that never been in humans at all, they are going to healthy volunteers first. So things are happening. There are many more trials and drug companies in this space than there were even a few years ago. So I think that is also something to be positive about. So the outlook has changed. We've gone from slow diagnosis, limited options for care, and no hope of clinical trials, and poor prognosis to better genetic understanding, proactive surveillance and wraparound care, and a small number of conditions with treatment-specific wins for those conditions, and more clinical trials and drug programs in development. And what I hope we're going to get to is earlier diagnosis, more precise therapies, curative treatments, and looking at trial outcomes that matter to patients and families. I think that one thing I do want to say here is that I've been looking after patients with mitochondrial disease for a large number of years, and we are transitioning a lot more patients from my pediatric clinic to adult life than we were in the past, even though we do not have curative therapies for most of those patients. And it is because we're getting better at monitoring and providing targeted supportive care. So that really is very important. I wanted to say a couple of things before I start on the wrap up, which is that I know that you as patients and families with individuals with mitochondrial disease, where they may be life-limiting, it's very urgent, you're desperate, and I don't blame you for being desperate. You're going to come across things. People might approach you. There are some bad actors in all walks of life. And so I would suggest that you have a conversation with your specialist team, and you can look for registered clinical trials, and official trials are listed in this website, clinicaltrials.gov. There's also an EU equivalent, and there's also a Japanese equivalent. And you want to look for evidence in people, not just cells or mice or social media. And you need to have a discussion of what the risks are, what the potential benefits are, what we don't know. Be cautious when you hear that there are dramatic claims or it's only lab data, or if you're being pressurized to pay very large sums of money up front, and promises of a personalized cure without independent specialist review, and any service that discourages you from checking with your NHS or specialist team. I would be very suspicious of that. Having said that, there's a lot that we are doing that's helping to develop clinical trials. So diagnosis has been improved. We are doing natural history studies. There is a natural history study that's ongoing for POLG disease and for Lee syndrome, and those have both been pushed forward and funded by the patient organizations, and we are all working in partnership here. I think that's really important to know. We need to understand the natural history of more than 400 different ultra-rare diseases, so that is challenging. And we also need better ways to measure the disease and the effects of treatments. So this is what we call biomarkers and outcome measures. I've told you that there are a lot of candidate therapies out there, that clinical trials are being done, and we are trying to engage all the multiple stakeholders involved here. I am putting this up. I know that you're all probably familiar with this, but all of those different problems I showed you in that first cartoon, there's probably something as doctors that we can do to try and address those problems. So supportive treatments are incredibly important. They improve quality of life and they may be life-saving. For example, pacing in someone with heart block because of mitochondrial disease. We're also working with patient organisations and through the European Reference Network to develop patient journeys for different types of mitochondrial disease. This is one of the early ones that we did for pyruvate dehydrogenase deficiency, and others are coming. And I've also been working with the Genomics England team on their newborn genomes program, which is called the Generation Study, which is embedded in the NHS. And that's sequencing the genomes of 100,000 newborns, but only for treatable genetic disorders. And within those massive, more than 400 mitochondrial diseases, some of them are treatable with vitamins or cofactors. And I have made sure that all of those are being screened for in this project. And also we're just about to get PDH added in because the ketogenic diet does make a difference for PDH deficiency. The 4 principles by which the Generation Study is founded upon are that you can reliably detect that gene, so you know that the variants cause disease, that it has a debilitating impact on quality of life early in life. We don't want to be diagnosing diseases that aren't going to present until you're 70 years old, that the outcomes are substantially improved by the treatments that are available, and that those treatments are universally and equitably accessible for everybody in the country. Okay, and then I do a lot of work trying to engage stakeholders, and they include, obviously, patient support organisations such as the Lilly Foundation, but also scientists, investigating mitochondrial disease, clinical researchers, clinicians, biostatisticians that we need to help design those trials, pharma companies and regulatory agencies, and most importantly, children with mitochondrial disease. And one of the things I think is really good about our country is that we do have these young patient fora and young patient advisory groups. So patients themselves can, when they're children, they can, not just adults, can give input into what is acceptable and not in a clinical trial. So to conclude, I hope what I've shared with you is that we are in a better place than we were even a few years ago. There are real treatment wins in a few conditions, but for most mitochondrial diseases, curative treatment is still some way off. But we are making progress in putting the science, the trial infrastructure, and the patient voice as strong, much stronger than they were before. And I truly believe that families deserve honesty, access to expertise, and reasons for hope. I don't want to have been here taking away your hope, but I also didn't want to be here giving you false hope. And that was a fine line to walk. As I said earlier, I'm very grateful to you for sparing your Tuesday evening to listen to me.